Does that in two places. First of all, this is the clamp, so there's the beta subunit. In prokaryotes, there's actually a donut like that. It's a dimer. So it's a beta 2 structure, and it forms a ring right around the DNA. You can see the template strand going right through the middle. This is the alpha subunit there. That's the guy that does the polymerizing, actually. In eukaryotic cells, there is a beta clamp. It's a trimer. It's a homotrimer. But anyway, there's the beta clamp. All of those other subunits that are in that diagram are involved in loading that on there. It does it 2 times, and the other instant, I don't know how to say this, it's involved in loading and also reloading. Remember I said when you reverse the strand like this, right, you can see you're synthesizing this fragment, and then you're going to have to let go and clamp down here and loop it around so you can synthesize the next fragment, right? It'll be the 3' here and start that. In fact, there's a primer. So you'll have to flip it around so that you can synthesize that piece of DNA in there on that primer. In order to do that, as soon as this guy, you can see this goes out, the one that's synthesizing now is going to grow until it meets here, and then it has to unclamp, let it out, let that strand flip around, and then clamp again. And all of those proteins that I showed you in that table are involved in opening this other clamp, letting the thing slip, and then clamping it again. That's obviously quite a process because it needs 5 separate subunits to do that. Don't see that? This is sort of one of my favorite guys here. This guy's called tau. What do they say tau is? 1, 2, 3, 4. Assembly processivity switch. I don't know why they call it that. Assembly processivity switch. I'm not sure where they get that name. But the tau protein sits right here between that. You notice this is an alpha beta epsilon. So this is the polymerizing unit. It's reading this template. This is another one that's reading the other template, the other strand. But there's a problem when you do this, right? And that is these guys are moving down this DNA as it's opening in this direction, right? It's moving down the DNA as a single unit. Well, what happens? Why do these two even stay together? Because if this is a lagging strand and this is loading and loading and loading, you would think that this one would take off and get far ahead, right? Well, what tau does, and I'm not sure why they get the name, but what tau does is it sits in here and it links these two together. And it gives them a little bit of jiggle. Right? They can jiggle by a nucleotide or 2, but they can't pull away from each other. So what it does is it slows this guy down to wait for this guy. So when this guy unclamps and he's reclamping and everything, since they're jiggered together like that, they sit together and wait, and then they go together again. So tau's kind of a fun protein. A couple other proteins in here that I should show you, I guess. In bacterial cells, there's this one called SSB. That's this green guy. They show it as a tetramer. It's a homotetramer. The protein's a homotetramer. And what it does is it binds single-stranded DNA. It's SSB, single-stranded binding protein. You can see wherever there's single strands, it's down there. And it's very important, right? Because when you melt DNA into single strands, and certainly when you have 1,000 nucleotides there, right? What happens if you have an inverted repeat? You form hairpin loops, right? You can form intra rather than inter. The hydrogen bonding between 2 DNA strands, intermolecular bonds, keeps them like this. But if you have a single strand, you can form intermolecular bonds between the single strand. And you can make a hairpin loop, right? You can design, I don't know what I did with that chalk. You can make a hairpin loop like this where there's bonds like this and it's all 5' and 3' end, right? And DNA polymerases, in fact RNA polymerases, all the polymerases that use templates are really very, very sensitive to structure. So if there's any sort of little bump, any sort of hairpin, they stop. They can't deal with it. So what single-stranded DNA does, SSB does, it keeps that single-stranded DNA from forming intramolecular hydrogen bonds and forming structures and hairpins or whatever that disallow the polymerases to work. That's SSB. What's DNA gyrase? Who knows what DNA? Oh, first of all, what's this guy here? So there's 2 things right here in this little complex. One of them is this guy here, which is a helicase. We've talked about helicases before. Helicase is actually an enzyme. It's a homohexamer. It has 6 subunits. They can all hydrolyze ATP. And the hydrolysis of ATP is what powers the unwinding of this DNA. It unwinds the DNA right at the template there. This little nugget there is a primase, leading strand template. What does it say? This is called primase. Both of these together are called helicase primase, right? And that's the primer, and what that guy does there is he actually builds the primer. So he puts these little primers on, he's put one up here at the beginning of the leading strand, and then every time it opens up 1,000 nucleotides, this guy there, which is right where it opens, adds another little one. So the primase puts on that little primer, and the helicase opens up the strand. So far, all of this stuff, all of these proteins are part of the replication complex. The polymerases, the helicases, the primers, the single-stranded binding protein, and other proteins. There's like 100 of them there. We're talking about a few. Does everybody understand these? This is called the primase complex, and it's part of this whole replication complex. The only other protein is gyrase. Who knows about gyrase? What's gyrase do? So what happens as this unwinds there? Remember, the DNA, the chromosome in this guy, E. coli, is a circle. And in your DNA, the longest chromosome in your body has a piece of DNA about this long, right? And so you're going to start unwinding it. And what happens as you unwind it like this? It gets tight, it gets taut, right? If you have a piece of rope that's braided and you tie it to that wall and you sit down here with your ATP and start to stretch it and pull it apart, and you first pull those strands apart, it's pretty easy. As you get up, it gets tighter and tighter. Eventually, it even flips and makes another coil, which makes it even harder to do. And you get to the point where you don't have enough energy to unwind anymore. Same thing here. It takes ATP energy to unwind those DNA strands, and the tauter it gets, the harder it is. Eventually, that 7.6 kilocalories of energy in ATP is not enough to break the hydrogen bonds because they're under such stress. So you have to relieve it. And the enzyme that does that is called gyrase. Gyrase is in the family of topoisomerases. There are two kinds of topoisomerases, right? I don't know if I have a picture of that or not. I don't have a picture, but there's two types of topoisomerases. Topoisomerase. There's 1 and 2. The difference is this guy makes a single neck. And this guy makes a double neck. I think I have a picture of this somewhere, I'll show you. But so what happens is the topoisomerase 1, gyrase is the type of topoisomerase 1. It makes a single nick, so it nicks one of those strands there, and when it does, the strands relative to each other can go whoop, and they unwind again, and puts them back together. Right? So it lets these guys twist and unwind so that there's no tension, and then the helicase can then unwind it. So that's what gyrase does. All of these are part of that complex that's leading that DNA synthesis. Question about these proteins? This is just a picture that shows you a donut. This is coli. You can see it's a homodimer DNA strand going through the middle. The molecules form that donut. I talked about that. So here's DNA ligase, and I'm not going to go tremendously into this activity. In E. coli, it's a nicotinic mononucleotide, and it's dependent, right, on, it's NAD dependent. And so essentially what happens is that here's the enzyme, and the enzyme has this amino group there, and that amino group becomes phosphorylated. It uses ATP to put a adenyl group on there, so adenosine monophosphate, right? There's the phosphate, there's the adenosine monophosphate is attached to that amide group on the enzyme, right? This amide, that free amide is the lysine, it's the epsilon lysine, it's the R chain on the amino, or the R on the amino acid lysine, right? So essentially, that guy becomes adenylated, and then what the enzyme does is it takes template. Here's the guy where we need the ligase, 3' hydroxyl, 5' phosphate, right? 3' 5' monophosphate. And what the enzyme does is it then takes this, it attaches this adenylate phosphate, right, onto this phosphate there. So there's the phosphate there, and here's the adenyl. There's adenine, right? Adenyl phosphate, so it's adenine monophosphate that's attached there. So now what that does is it gives us this phosphoester bond in there, that PP, the phosphoester. That phosphoester bond is a high energy bond. So now what the ligase can do is sort of work like a DNA polymerase, right? This hydroxyl group can attack that phosphoester bond there, and it can form it forms a phosphoester between the hydroxyl group and that phosphate. And when it does, this group there is then released back. So there's the AMP coming off, the adenylate group, the adenine monophosphate coming off, right? And the enzyme goes back to where it was. You see that? It's sort of complicated. You can look at it, but it does what I'm telling you, and that is when you have this thing that you need to ligate, that gap in there, there's no energy because this monophosphate has no energy. There's no energy in that that ester linkage there. So what you do essentially is generate a diphosphate. You generate a pyrophosphate that has that phosphoester in there that's an energy, containing bond. Now you can do the biochemistry of this hydroxyl group and undergo a nucleophilic attack on that diphosphate and form your phosphoester. Okay? Question? That's the hardest thing we've done today. Look at this, and if you have a question, ask me, because it's easy to explain. Okay, what I want to start on, we don't have a lot of time, but I want to start on how DNA initiates, and the initiation of DNA synthesis. We talked a little bit about DNA synthesis minimally. And if you have questions, you should ask about it. Your book has lots about it. So the question is, where does DNA synthesis begin, and how does it begin? And in E. coli and bacterial cells, and actually it occurs in eukaryotic cells as well. But in E. coli, remember the chromosome is a circular chromosome. There's that circular chromosome. And there is a site in there that's called the origin of replication. DNA replication always begins on the same site. And if you look at that origin of replication, if you blow it up, what you see is there are repeats in there. There's two sets of repeats. One has 3 repeats in it, and the other has 4 repeats in it. And if you look at this, these 4 repeats, there's 9 base pairs, and these guys have 13 base pairs. Now that 9 base pair repeat is a repeat that's recognized by a protein that's called DNA. Proteins that are involved in DNA synthesis are called, you can see there's one that's called DNA A, there's one called DNA B, there's one called DNA C, one called DNA T. We're going to talk about just a few of those. But DNA A is a protein that recognizes that 9 base pair repeat and binds to it. And the binding of DNA A to that repeat is cooperative. What that means is that when one binds, the other binds more quickly, and the third one even more quickly than that. And not only, once they start binding, they interact with each other, they interact with that DNA. You can see that what happens is they form this clump of these molecules here, or these proteins bound to that DNA, and they actually clump around that binding site, around those repeats, and they form, they do this to the DNA. And notice what they've done. They made a knot. They kind of looped this DNA. They sort of put a knot there. So first of all, they recognized the origin, right? They found it. And second of all, they're doing this twisty thing to make that knot. What do you think that might be doing there? What do you think that might be doing? To take that DNA and twist it around in a knot. Think about the gyrase story, right? So when you take, you can do it with a piece of rope, but what's happening is as you twist this around, this strand of double strand of DNA, right, you're putting tension on this region down here. Right? The more you twist it, the more tension you're putting down here. So what that does is it gives this region here the tendency to begin to sort of